Have you ever stopped to think about how you can digest a meal, or blink your eyes, or even just think? I mean, really, think about it. It all comes down to these incredible microscopic machines working non-stop inside every single one of your cells. Today, we're doing a deep dive into the unsung heroes that make all of life possible: enzymes.
So, let's kick things off with a little puzzle. Your body, at its core, is just a big bag of chemical reactions, right? But if you've ever been in a chemistry lab, you know that most reactions need a ton of heat or pressure to get going. So how on earth are you running all of these complex reactions perfectly fine at a pretty mild 98.6 degrees Fahrenheit? The answer, my friends, is both elegant and powerful, and it's exactly what we're here to talk about.
All right, Section 1, Life's Ultimate Accelerators. Let's get right to it and figure out what these biological catalysts actually are.
Okay, so here's the textbook definition, but let's break it down. An enzyme is basically a biological catalyst. Its one job is to speed things up, and I mean speed things up by millions of times. The secret sauce is how it lowers something called activation energy. You can think of activation energy as the huge push a giant boulder needs to start rolling down a hill. Well, enzymes give that push, a really big one, allowing reactions to happen super fast at just normal body temperature. And you know what the coolest part is? After the job is done, the enzyme just pops right out, completely unchanged, and is ready to go again. Incredible!
So that brings us to Section 2, inside the enzyme machine. How does a simple string of molecules turn itself into a super-specific, complex 3D tool? Well, it all comes down to one amazing process: folding.
It happens in a few key stages. First, you have the primary structure, which is really just the sequence of amino acids, kind of like letters in a word. Then, in the secondary stage, that chain starts to twist and pleat into cool shapes like coils and sheets. But the real magic, the main event, happens at the tertiary stage. This is where the whole thing folds up on itself into a very specific, very complex 3D shape. For a lot of enzymes, this is it. They're ready to work. Some of the more complex ones go one step further to a quaternary structure, where a few of these folded chains team up to form a superenzyme.
And this right here is the absolute crucial point. That specific 3D folding is not random at all. It meticulously creates a special little pocket or groove, and we call that the active site. This is the business end of the enzyme. It's where all the action happens, and its unique shape is, well, it's everything.
So what's holding this ridiculously precise 3D shape in place? It's a whole combination of different chemical bonds. And to help you remember them, just think of the word HIDs. You've got hydrogen bonds, ionic bonds, super strong disulfide bridges, and something called hydrophobic interactions. Think of these bonds as the microscopic scaffolding that maintains the perfect shape of that active site. And as we're about to see, if you mess with these bonds, you mess up the entire enzyme.
Okay, Section 3, how enzymes get to work. So we built the machine, we know what it's made of. How does it actually do its job? Well, it's like an elegant little dance between the enzyme and its target molecule, which we call the substrate.
You know, for a long time, scientists went with something called the lock and key model. The idea was simple. The substrate fit into the active site perfectly, just like a key slides into a lock. It's a nice image, but we now know it's not the whole picture. The current, much more accurate model is the induced fit model. In this version, the active site is actually a bit flexible. When the substrate comes along and binds, it actually causes the enzyme to change its shape ever so slightly, creating a perfect, snug fit. And this little hug is brilliant. It not only holds the substrate tight, but it also puts stress on its chemical bonds, which helps the reaction happen. And hey, for anyone studying for an exam, induced fit is the answer you're looking for.
The whole process is this beautiful, efficient cycle. The substrate just bumps into the active site and binds, and for a brief moment, they form an enzyme-substrate complex. That's when the induced fit happens. That activation energy gets lowered, and boom, the reaction happens. The substrate is converted into new things, the products, and then they're released. And just like that, the enzyme's active site is free, totally unchanged, and ready for the next customer. It is just so efficient.
But our enzyme hero isn't invincible. Just like any high-performance machine, it has its ideal working conditions. So let's take a look at the environmental factors that can either make it work perfectly or break it completely.
First on the list is temperature. And really, it's a bit of a Goldilocks story. If it's too cold, things are sluggish, the reaction is slow. As things heat up, the reaction rate gets faster and faster, until it hits a perfect sweet spot. That's the optimum temperature. But if you go too hot, the activity just plummets. It falls off a cliff.
And the reason why this happens is super important. As you turn up the heat, all the molecules start moving faster, right? That means more energy and more frequent collisions between the enzyme and its substrate. That's why the rate goes up. But if you go past that optimum temperature, all that extra heat energy starts to vibrate the enzyme so much that it breaks those delicate HID's bonds holding its shape together. The active site changes shape permanently. We call this process denaturation. And at that point, the enzyme is basically broken.
And what's really interesting is that we see a very similar story for pH. Every single enzyme has a specific pH level where it works best, its own unique optimum. For example, the enzyme pepsin in your stomach absolutely loves highly acidic conditions, but an enzyme like trypsin in your intestine needs an alkaline or basic environment to do its job.
The reason for this is all about the chemistry. If the pH gets too acidic or too alkaline, it starts messing with the electrical charges on the amino acids, and that, in turn, disrupts the ionic and hydrogen bonds. You know, part of our HCDS scaffolding that hold the enzyme's critical 3D shape. So just like with too much heat, the active site changes shape and the enzyme just can't work properly anymore.
And hey, for anyone studying this stuff, here's a fantastic little mnemonic to remember all the key factors that affect how well an enzyme works. Just remember TPSEI. That's temperature, pH, substrate concentration, enzyme concentration, and finally, inhibitors. Super, super useful.
Right, on to section 5, inhibitors. That last letter in our mnemonic, I, stands for inhibitors. And if enzymes are the heroes of our story, then inhibitors, well, they're the villains. These are molecules that sabotage the whole system by getting in the way of the enzyme's work.
Now, there are two main types to know. A competitive inhibitor is a bit of an imposter. It looks a lot like the normal substrate, and it literally competes for the exact same active site, physically blocking the real substrate from getting in. A non-competitive inhibitor is a bit sneakier. It doesn't bind to the active site at all. Instead, it binds to a totally different spot on the enzyme, which causes the active site to change its shape from a distance. Now, here's the key difference. You can actually beat a competitive inhibitor by just flooding the system with more of the real substrate. But with a non-competitive one, adding more substrate won't help one bit.
And of course, we've got another great mnemonic to help you lock this in. Just remember the word cannot. C-competitive binds to the active site, and non-competitive is not overcome by more substrate. It's a really simple trick to keep them straight.
All right, section 6, enzymes in our world. We've been deep down in the molecular weeds for a while. Let's zoom back out and see how these tiny little machines have a massive impact on everything from what's happening in your gut right now to the products you buy at the grocery store. The short answer is, they are absolutely everywhere. Right now, inside you, enzymes like amylase and lipase are busy breaking down your lunch. Meanwhile, in your laundry room, the very same types of enzymes are in your detergent, literally eating away grass and food stains. And in medicine, they're the key ingredient in things like blood glucose monitors for people with diabetes, acting as these incredibly specific little biosensors.
Now, to make enzymes even more useful for big industries, we often use what are called immobilized enzymes. Basically, we attach them to a solid surface, like tiny little beads. This simple trick makes them more stable, makes it super easy to separate them from the final product, and, this is the big one, it lets us reuse them over and over again. It makes these huge industrial processes way more efficient and a whole lot cheaper.
There's a great quote that says, "Enzymes are not only the catalysts of life, but also the catalysts of industry." And it's just so true. They are the fundamental little engines driving all of biology, and we've gotten really clever at harnessing their power to drive our own modern technology too.
And that really brings us to the end of our journey today. We've seen that enzymes are these specific, efficient, and incredibly powerful molecular machines. So that leaves us with one final big thought. If enzymes are basically nature's programmable nanomachines and we can already use them to wash our clothes and monitor our health, what are we going to build with them next? As our ability to design and engineer proteins gets better and better, what new problems will we solve with nature's ultimate accelerators? Thanks for joining me on this one,